Hello, my dear students. I welcome you all to another video lecture, and that is going to be about the structure of skeletal muscle. Now, what we are going to do is we are going to have a muscle tour. We'll go from a muscle belly deep inside the basic and the smallest contractile unit and the functional unit of the muscle cell, that is sarcomere, and we will see we will go up to the level of the myofilaments, that is the actin and myosin. Okay, let's get started. So the contents for today's lecture, we'll have the introduction to the skeletal muscles, and then we will talk about the properties and functions in brief. Then we will talk about the organization of skeletal muscles, and then we will talk about the structure of sarcomere. Okay, that is, the sarcomere is the basic functional and the smallest contractile unit of the skeletal muscle. Now, in introduction, we all know that the skeletal, we have the three types of muscles present in our body. We have the skeletal muscle, we have the cardiac muscle, we have the smooth muscles. Now, cardiac muscle is present in the cardiac tissue, in the heart, and then the smooth muscles that actually are present in the visceral organs, in the internal body organs, and then the skeletal muscles which are attached to our skeleton, which are attached to our bones, that is the skeletal muscles. Now, skeletal muscles, because these skeletal muscles are in our own control, we call them as the voluntary muscles. Okay, so that is the first thing you have to understand, that is, these skeletal muscles are the voluntary muscles. These are the voluntary muscles. That means these skeletal muscles are going to be in the control of the somatic nervous system. Okay, because we have two types of the or two divisions of the peripheral nervous system. One motor peripheral nervous system, one is the autonomic nervous system, and second is the somatic nervous system. And somatic nervous system controls our voluntary actions, and that means that this somatic nervous system is going to be controlling our voluntary movements, that is the skeletal muscles. The second thing is that these muscles, just like the cardiac muscles, these are the striated muscles. These show the striations. There are the lines that are appearing in the structure of the skeletal muscles as well as the cardiac muscles, but those striations are not present there in the smooth muscles. We will talk about these striations as well. Okay, now, as I've already told you, that these skeletal muscles are attached to our bones. They are attached to our skeleton, and the attachment is provided to the bones. The attachment of these skeletal muscles to the bones is provided by a special connective tissue that is called as tendons. Now, this is mainly by the tendons, and it is also provided by aponeurosis, or there can be the direct attachment to the bones as well. Then these cells, these skeletal muscle cells, when we will see in the diagram, here also you can see that these skeletal muscle cells, they are long cells, they are the long cylindrical cells. So in order to fulfill that demand or the formation of proteins inside the cell, one nucleus was not going to be enough. So we need multiple nuclei for the formation of proteins inside the muscle cell. So these skeletal muscles are multinucleated cells. And last thing you have to understand in the introduction is that these skeletal muscles are specially arranged to generate the force. They are specially arranged for the force generation. We will talk about all these things in the lecture. Now, when we talk about the properties and functions of skeletal muscle, there are four important properties that you have to understand and know about. The first property is excitability. Excitability is that they get excited. They have the ability to respond to a stimulus. For example, a motor neuron is coming from the central nervous system, and that motor neuron is going to stimulate the muscle cell. the skeletal muscle cell, and the skeletal muscle cell responds to that stimulus and generates the action potential. That property is called as excitability. The second thing is contractility. When it responds to that stimulus, when this skeletal muscle cell, it responds to that stimulus, it generates the action potential, and ultimately what is going to happen, that this muscle cell is going to contract. It is going to contract, it is going to get short. That is the contraction. So that property is called as contractibility. Then we have the extensibility. It is the stretching of the muscle beyond the normal resting length of the skeletal muscle. So that stretching, it can get stretched, the skeletal muscle. So that is the stretchability, extensibility or stretchability. And the last property is elasticity. After getting stretched, okay, beyond the normal resting length of the skeletal muscle, or after contraction, that is shortening of the length, this is going to go in the normal resting length after that. So that is called as elasticity. That means it is going to, after stretching, it is going to recoil back to its normal resting length. That is called as elasticity. And when we will talk about in the future lectures about the the smooth muscles do not have the property of elasticity, because the proteins that are providing this elasticity to the skeletal muscles are not present there in the smooth muscles. Now, when we talk about the functions of the skeletal muscles, they are very important in producing the movements. I've already told you that these skeletal muscles are attached to the skeleton. So they are attached to our bones. Now these skeletal muscles have the first very important function, that they are very important in producing the movements. When these muscles contract, they pull the bones they are attached to, and that leads to the movement of that skeleton, movement of these bones. The second thing is they are maintaining the posture. They are very important in maintaining the posture. If I am standing right here in front of you delivering this lecture, maintaining my standing posture thanks to the skeletal muscles. If you are sitting upright and listening to my lecture and maintaining your sitting posture, again thanks to the skeletal muscles. So these skeletal muscles are very, very important in maintaining the posture of our body. Then they are very important to provide that stability to the joints. And last but not the least, they are very important in producing or generating the heat in our body. I hope you remember that lecture of homeostasis. Homeostasis mein we talked about the negative feedback mechanism. When our body temperature is dropping, when our body temperature decreases, our body tries to increase the contraction of the skeletal muscles and producing that shivering. And that shivering is the contraction of muscles, and during the contraction of muscles, heat is produced there as a byproduct. So there is the production of heat. So they have the role in maintaining the temperature in our body. So they are very important in the thermoregulation in our body. Then we will talk about the organization of the muscle. Now, what we will do is we will have a tour of the muscle, deep inside the muscle, right from the muscle belly up to the myofilaments, that is the actin and myosin. They are doing all the drama inside the muscle. Now, when we talk about the muscle, for example, we will talk about the biceps muscle. Okay, so bicep muscle jo hai, we will take a chunk of this bicep muscle, we will take a slice of the bicep muscle, and we will see what are the structures we are going to see inside. Now, inside this muscle belly, we definitely have the muscle cells, we have the muscle fibers, and those muscle cells and muscle fibers are arranged in bundles inside. Okay, so this is the muscle belly, and inside the muscle belly we have, these are the small jo aapko dikh rahe hain, these are the muscle fibers, they are the muscle cells. But you can see in the diagram that these muscle fibers or muscle cells are arranged in these bundles, and these bundles of muscle fibers are called as fascicles. Okay, then we have inside the fascicle we have these muscle fibers. Now muscle fibers may, when we see inside the muscle fiber, we have a structure, something called as the myofibril. Okay, myofibrils are the rod-shaped structures that are present inside the muscle cell. And when we talk about inside the myofibril, when we take this tour deep into the myofibril, we see something called as the myofilaments. And myofilaments are present in this structure that is called as sarcomere, and this sarcomere is present inside the myofibril. What did I tell you? Myofibril is a rod-shaped structure. For example, consider this myofibril as a train, and the compartments of this train are going to be the sarcomere. Okay, and that sarcomere is actually what is the smallest contractile unit of the skeletal muscle. Okay, and inside the sarcomere we have these myofilaments, the proteins that are actually responsible for the contraction of muscles, and those filaments are the actin and myosin filaments. Okay, now let's talk about all these things one by one. Now let's start with the first thing, the muscle belly. When we talk about the muscle belly, this muscle belly is surrounded by a connective tissue. For example, this is a muscle belly and it is surrounded by this connective tissue. So outside this muscle belly, we have a connective tissue, and that connective tissue covering is called as epimysium. That connective tissue covering is called as epimysium, and this epimysium is the dense, fibrous, irregular connective tissue. So that means it is going to be very tough. So it is very tough and it is made up of dense, irregular connective tissue. So that means it is going to give that protection and strength to the whole muscle belly. Okay. Now, after that, what did I tell you? That inside this muscle belly, we have all these, inside the muscle belly, we have the bundles of these muscle fibers, and these bundles of these muscle fibers are called as fascicles. That I've already told you. This is a bundle of muscle fibers and this is called as a fascicle. Now suppose we are pulling one fascicle out of this muscle belly. Here is one fascicle. Here is the bundle of these muscle fibers. And this fascicle also is covered by a connective tissue layer or by a connective tissue covering, and that covering of connective tissue is called as perimysium. Okay, now the perimysium is also the dense, fibrous, irregular connective tissue. But remember that this perimysium is continuous with the epimysium. Okay, so when the perimysium is pulled, it pulls the epimysium as well. Okay, now inside the fascicle, we have these muscle fibers. Okay, and this muscle fibers, for example, this is a fascicle, and fascicle is covered by a connective tissue layer that is called as perimysium. Now we have pulled one muscle fiber out of this fascicle, because I've already told you that fascicle is nothing but a bundle of muscle fibers. Now pulling one muscle fiber out of this fascicle, this muscle fiber is also containing one connective tissue layer that is surrounding this muscle fiber, and that connective tissue layer is called as endomysium. But this endomysium is not as tough and strong as the perimysium and epimysium, because this endomysium is made up of loose areolar connective tissue. Okay, this endomysium is present outside the sarcolemma. Sarcolemma is the plasma membrane or the cell membrane of the muscle fiber, of the muscle cell. Suppose this is the muscle cell, and this muscle cell has this cell membrane, and this cell membrane is called as sarcolemma. Now outside this sarcolemma there is the connective tissue layer, and that connective tissue layer is called as the endomysium. So epimysium covering the whole muscle belly, the perimysium covering the fascicle, the bundle of muscle fibers, and endomysium covering one single muscle cell outside the sarcolemma. But this endomysium is not just outside the sarcolemma. There is something that is actually in between, these this जो है endomysium and the sarcolemma, and that is the basal lamina. Okay, that is the thin sheet of connective tissue, actually the extracellular matrix of the muscle fiber. Okay, now suppose here we have this muscle fiber, and this muscle fiber, and here is the endomysium. Okay, endomysium is here, and endomysium or muscle fiber के sarcolemma इसके बीच में there is the basal lamina. Now basal lamina, please understand this, basal lamina और इस plasma lemma या sarcolemma के बीच में there are spatial stem cells present, and those stem cells are called as satellite cells. Now what are these satellite cells? These are the stem cells, and they are very, very important for the repair and regeneration of muscle fibers. Now for example, there is any tear to the muscle, there is any injury to the muscle, we need that repair and regeneration of muscles, and that is provided by the satellite cells, and these satellite cells are sitting in between the plasma lemma or sarcolemma and the basal lamina, and outside that basal lamina there is the endomysium. Okay, now going back here, we have after endomysium, what did I tell you? That this whole muscle is connected to the bone, and it is connected to the bone. The attachment is provided by a special rope-like connective tissue that is called as tendon. Okay, and this rope-like connective tissue is rich in collagen. Okay, it is rich in collagen, and this connective tissue rich in collagen, that is called as the tendon, it gives the attachment of the muscle to the bone. But it is not the only thing that provides the attachment. For example, there is another thing. There is a sheet of connective tissue that is providing this attachment of the muscle to the bone, and that sheet of connective tissue is called as aponeurosis. Okay, so aponeurosis is also providing the attachment of muscle to the bone, but mainly the attachment is provided by the tendon, and the tendon is the rope-like structure this protein is having, and aponeurosis is the sheet-like जो है connective tissue. Now, one more important thing is that sometimes there can be the direct attachment of this muscle with the bone or cartilage. Okay, so epicondrium or periosteum or perichondrium जो होता है, that is the layer of the bones and the cartilage, उसके साथ भी direct connection हो सकता है muscles का, but mainly the connection is provided by the tendon, the connection between the muscle and the bone. Now, moving forward, we have to talk about the muscle fibers. Now, muscle fibers are the long rod-shaped structure or cylindrical. They are the long cylindrical and multinucleated cells. Now, these muscle fibers also called as muscle cells. So, whenever you hear the word muscle fibers, it is nothing but the muscle cells. And these muscle cells are present in the form of bundles, and those bundles we have already talked about are called as fascicles. Now, we are talking about one muscle cell. And the muscle cell is multinucleated, because I have already told you that these are the long cylindrical cells. Okay, and we have to be involved in the formation of proteins all over the muscle cell. So one nucleus was not going to enough. So we have the multiple nuclei that are present in the muscle cell. Now, this muscle cell or muscle fiber, it is having the cell membrane. And that cell membrane, as I've already told you, that cell membrane is called as sarcolemma. Okay, that is called as sarcolemma. And the cytoplasm inside the muscle cell or muscle fiber, the cytoplasm, for example, that cytoplasm is called as sarcoplasm. sarcoplasm, and this sarcoplasm contains all the cell organelles, most importantly the mitochondria. Then it also includes it also contains the myoglobin. That myoglobin is that oxygen binding molecule there. It also contains glycogen, the storage form of glucose, because we need all the glucose there inside the muscle cell for generation of the ATPs or energy inside the skeletal muscle. Okay, now moving forward, this is the sarcolemma, this is the or the plasma lemma, this is the plasma membrane or cell membrane of this muscle fiber or the muscle cell. Now in this plasma membrane or the sarcolemma, there are invaginations, and those invaginations, for example here, these invaginations are making the tube-like structures here. They are going deep. These invaginations in the cell membrane, they are going deep and they are making a tube-like structure, and that tube is called as T-tubule. Okay, you are going to know about the importance of this T-tubule when we will talk about the physiology of muscle contraction. But here I am telling you that there are invaginations in the cell membrane, and these invaginations are forming the tube-like structure, and this tube-like structure is called as T-tubule. Now, this skeletal muscle has to go for muscle contraction, and you might have an idea that there is an important, very important role of calcium in the muscle contraction. Now that calcium is stored inside the cell in a cell organelle, and that cell organelle is going to be sarcoplasmic reticulum. Okay, now it is going to be sarcoplasmic reticulum. So sarcoplasmic reticulum is highly developed inside this skeletal muscle. Now sarcoplasmic reticulum, it is the specialized smooth endoplasmic reticulum which is present in every cell, but it is highly developed and it is named as sarcoplasmic reticulum in case of skeletal muscle. So sarcoplasmic reticulum has the structures, and these structures are called as terminal cisternae. These structures are called as terminal cisternae. For example, here is the sarcoplasmic reticulum. It has the terminal cisternae here. Then there is the T-tubule. Here is the sarcoplasmic reticulum. It has the terminal cisternae here. Now, we can see in the diagram that there is the T-tubule, and T-tubule have on both sides these terminal cisternae of the sarcoplasmic reticulum. Now, all these three things together, two terminal cisternae, in the middle there is T-tubule, we call this structure as a triad in the skeletal muscle. Okay, this is very important. Now, moving forward, we have to talk about the myofibrils. I have already given you the example of a train. Okay, for example, the muscle fiber, inside the muscle fiber we have the rod-shaped structures. We have the rod-shaped structures, many rod-shaped structures, and those rod-shaped structures we call as myofibrils. And I've already given you the example. Consider these myofibrils as a train. So this is a long train, and these myofibrils are surrounded by the sarcoplasm. Okay, that is the cytoplasm of the skeletal muscle. Okay, it is surrounded by the sarcoplasm, and I've already told you that sarcoplasm contains the glycogen, it contains all the mitochondria and cell organelles, and it contains the myoglobin as well. Okay, so now inside this train there are different compartments. Okay, inside this myofibril, for example, this is the myofibril, and this myofibril inside has many compartments. If this myofibril is a train, there are the train compartments inside. Okay, and those compartments we call as the sarcomere. And the sarcomere is the smallest and the functional unit of the skeletal muscle. Okay, now the sarcomere, for example, here is this is the long train, and this compartment starts from here. Okay, this compartment starts from here and it ends here. Okay, so there is the zigzag pattern of this structure here, and these zigzag patterns we call as Z lines. We are going to see the Z lines in a little more detail in the upcoming slides. But here I am telling you what is this compartment actually, where this compartment is having the boundaries, and those boundaries are the Z lines. Okay, now let's move forward. Inside this myofibril, now let's have a look inside the myofibril. Inside the myofibril, for example, this is the myofibril whole structure. This is a rod-shaped structure, and inside this myofibril, what we are going to see is we are going to see the myofilaments. And myofilaments are of two types mainly. That they are the thin actin filaments, and we have the thick myosin filaments. You can see here, these red-shaped structures are the myosin filaments. They are thick filaments, and these blue-shaped structures we have, they are called as the thin actin filaments. Okay? Now, let me repeat. We have the muscle cell, and muscle cell, or we call that as the muscle fiber. Now, inside the muscle cell, we have the rod-shaped structures. We call them as the myofibrils. Sorry, myofibers. अब हमारे पास जो myofibril, and myofibril के अंदर जो है, we have the myofilaments, and those myofilaments are the actin and myosin filaments. Now, I've already told you in the previous slide that I've already told you in the previous slide that inside this myofibril, what is there is that it has the compartments, and these compartments are, one second. Okay, inside this myofibril, inside this rod-shaped structure, we have these compartments, and these compartments are these zigzag lines, and these zigzag lines are going to be the Z discs. We have the Z disc here, and we have the Z disc here. And from one Z disc or Z line to another Z disc or Z line, this whole structure is called as the sarcomere. I'm repeating this because this sarcomere is the basic functional unit and the smallest contractile unit inside the cell. Okay, now let's talk about the sarcomere in a little more detail. Inside the sarcomere, what did I tell you? It has the boundary on both the sides, and that boundary is called as the Z line or the Z disc. Okay, actually these names were given by the German anatomists in the past. So this Z line has the origin, German origin. It actually indicates the Zwischen. It means the Zwischen, and Zwischen actually means boundary. So this Z has been taken from the word Zwischen. It is the German word, and this Zwischen means boundary. And this boundary, because this Z line is providing the boundary on both the sides of the sarcomere, so this is called as the Z line. Now this Z line contains the spatial protein, and this Z line contains the protein that is called as alpha-actinin. This Z line contains the protein that is called as alpha-actinin. Now, why is this Z line important? Because it provides the boundary to the sarcomere, and second thing is it provides the attachment to two important proteins present in the sarcomere. One is it provides the attachment to these thin filaments. We call them as thin actin filaments. So you can see here, these are the thin filaments, and this thin filament is attached to this Z line. The second thing is that this Z line provides the attachment to this springy protein, this yellow protein here, and this springy protein is also attached to the Z line, and this springy protein is called as the titin. Okay, and it is the elastic protein. I have already told you in the properties of the skeletal muscle that the skeletal muscles have the property of elasticity. Okay, and that elasticity is provided by this protein that is called as titin. And what did I tell you? That the elasticity is not present there is not the property of the smooth muscles, and because the smooth muscles does not contain this protein that is called as titin. So this titin is anchored or it is attached to the Z line, and another protein that is attached to the Z line, that is these thin actin filaments. Okay, now let's move forward. We have the filaments inside the sarcomere. As I've already told you that there are two important filaments. One is the thin filament and second is the thick filament. Okay, now the thin filament are the actin filaments. Okay, these blue proteins, and thick filaments are the myosin filaments, these red thick jo hai filaments that you can see in the diagram. So we have two important filaments, myofilaments present in the sarcomere. We have the actin filaments and we have the myosin filaments. Okay, now let's talk about them one by one. The first is the thin filaments. So thin filaments जो है actin filaments, as I've already told you in the previous slide, that this actin filament is anchored or it is attached to the Z line, and the myosin filaments are attached to the titin, that elastic protein, and this titin is attached to the Z line. So this myosin is not attached to the Z line directly. The attachment is provided by the titin. So myosin is attached to the titin and titin is attached to the Z line. Okay, so remember here. This is attached to the Z line here. Now, so when we talk about the actin is actually made up of the actin monomers. We call that as G-actin. You can see here is the actin monomer. Here is one actin monomer, and this actin monomer is called as G-actin. G stands for globular actin. Okay, now this G-actin goes for the polymerization. So it gives the shape to the polymer jo hai of G-actin, and that polymer is called as the F-actin. And F-actin is called as filamentous actin. Okay, you can see here we have the G-actin, that is globular actin. G stands for globular, this is the monomer, and then when these monomers combine together, they form the polymer of actin or polymer of the G-actin, that is called as the F-actin. Okay, now this F-actin jo hai polymers of G-actin, they actually give rise to the supramolecular helix. Okay, they give rise to the supramolecular helix. You can see here, this is the helical structure. Okay, this is the supramolecular helical structure, and that actually makes our thin actin filaments. Okay, now in the actin, these actin jo monomers hai, they are making the polymer of actin. These actin monomers have the binding sites for the myosin. They have the binding sites for the myosin, and that binding sites to the myosin provides the attachment to the myosin, because that it is the binding site for myosin only. So it is the myosin that is going to bind with actin on this binding site. And you all might have an idea that this attachment of the myosin and the actin will ultimately lead to muscle contraction. Okay, iske baad there are some other proteins that are present in this actin thin filaments. We have the tropomyosin and the troponin. Now, when we talk about the tropomyosin, look here please, this is the actin thin filament, and this actin thin filament has here another protein, and that protein is called as tropomyosin. Okay, now what this tropomyosin does is it blocks the binding of myosin with the binding sites of actin for myosin. Okay, or simply it prevents the attachment of myosin with the actin. Okay, this tropomyosin. Now another protein we have there, that is called as troponin. Okay, and this troponin present there has 3 active sites. For example, here is the troponin. It has the 3 active sites, and the 3 active sites are, one is troponin C, and troponin C is the active site for the calcium. Calcium will come and bind with the troponin because the calcium has an important role in the muscle contraction. The second thing is troponin T, and this troponin T is the binding site for the tropomyosin. Okay, and third is troponin I, and it is the binding site for the actin. Okay. Now, why is this troponin I? Why it is called as I? Why not troponin A as it is for actin? Because this site, once it is bound to actin, it is the inhibitory site. Actually, it keeps the actin inhibited, or when this troponin is bound to actin, it is usually in the relaxation phase. Once it is bound to myosin, that time it will not be bound. So troponin I जो है, it is the inhibitory जो है site for the troponin. Now, next is one more important thing. Along with this actin, there is another protein, and that protein you can see here, and that protein is called as nebulin. And this nebulin is very, very important to maintain the stability of this thin actin molecules. Actually, it determines the length of this thin actin filaments. Because in all the sarcomeres, the length of the actin has to be constant in order to make that proper muscle contraction. So that length is of the actin filaments is determined by this protein that is called as nebulin, and this nebulin is running along the actin there. Okay, now let's talk about the next filament, that is the myosin. Now myosin जो है, it is composed of 6 polypeptide chains. Okay, it is made up of 6 polypeptide chains when we talk about the structure of the this myosin molecule. Now out of the 6 polypeptide chains, it has the 2 heavy chains and it has the 4 light chains. Okay, now you can see here it has the 2 heavy chains. These are the heavy chains here. Let me change the color, please. Okay, it has the 2 heavy chains here, and these heavy chains are making the tail, and ultimately these heavy chains turn here and they make this structure here that is called as the head of the myosin. Okay, so these are the 2 heavy chains, and it has the 4 light chains. And those light chains, they can be the regulatory light chain and they can be the essential light chain. A regulatory light chain is actually controlling, it is actually regulating the function of myosin or regulating that the head of the myosin. And जो ये essential light chain जो है, it maintains that structure of the head and the tail, and that is the essential light chain. And regulatory light chain जो है, it contains certain enzymes and it controls the function of the myosin basically. Now, here you can see we have the molecule, that is the myosin molecule. Here you can see there are the 2 heavy chains and there are the 4 light chains here. And once these many myosin molecules, they combine together and they form that filament, that is the thick filament, that is the thick myosin filament. And these myosin filaments जो है, they will attach to the thin actin filament, and that will lead to the muscle contraction that we will see in the upcoming lectures. Now, here is another protein that is very important, that is present in this muscle fiber, and that protein is called as dystrophin protein. Okay, here is the dystrophin protein. Now, what this dystrophin protein does is it connects the cytoskeleton of the muscle fiber. For example, the actin is here. It connects this actin with the जो है membrane proteins, and those membrane proteins are called as dystroglycan or sarcoglycans. So now these membrane proteins, what they do is they are connected with the extracellular matrix through another protein that is called as laminin. Okay, so now what this dystrophin does is, this dystrophin actually makes a connection, or it helps to connect the cytoskeleton of the muscle fiber with the extracellular matrix. Now, what happens is this dystrophin actually, while the contraction, it maintains that structural integrity of the muscle fiber. Okay, now there is a clinical condition. There can be this dystrophin protein is the gene responsible for making this dystrophin protein is dystrophin gene, and that dystrophin gene is present in the X chromosome. Now, X chromosome. Now, there can be mutation in the X chromosome, mutation in that gene, dystrophin gene. Now, what will happen? That can lead to the absence of the dystrophin protein. Okay, that means the dystrophin protein will not be formed. And when this dystrophin protein will not be formed, so what will happen during the contraction? That structural integrity will not be maintained, that strength will not be provided to the muscle cell. What will happen? There will be rupture in the cell membrane, there will be rupture in the sarcolemma, and that rupture will lead to the damage to the muscle cell, and that will lead to dystrophy. That will lead to dystrophy. And if this dystrophin protein is absent, that is called as Duchenne muscular dystrophy, DMD. But if it is formed but not functional, or it is formed less than normal, that leads to a type of dystrophy that is called as Becker muscular dystrophy. That is DMD and BMD. Okay, that we will talk about the muscular dystrophies in the upcoming lecture. Now, here we have in between two Z lines, in the middle of the sarcomere. In the middle of the sarcomere, for example, this is the sarcomere, and sarcomere has one Z line here and one Z line here. In the middle, we have the structure. It is a three-line structure there, and we call this as M line. And this M line actually holds all the myosin molecules or myosin filaments together, and they act as the shock absorber. Okay, they act as shock absorber, this M line, and it is present in the middle of the sarcomere. And this M line is made up of the proteins. Most important protein present there is the myomysin. Now, why M line? It also has the German origin, and this M actually has been taken from the word Mitte. And Mitte means middle, so it is in the middle of the middle of the sarcomere. So that means it is called as the Mitte line, or in short, it has been given the name as M line. Now, after that, we have, for example, this is the myofibril here, and this myofibril jo hai, I've already told you that it has these Z lines, it has these sarcomeres inside. But when we see this myofibril under the microscope, or when we see the muscle fiber under the microscope, we see all these striations, we see all the lines appearing in the structure of the muscle fiber or this myofibril. Okay, and these lines are called as the striations. Now we will talk about why are these striations there in the muscle fiber or the myofibril. The first thing you have to understand that this sarcomere or this myofibril has been divided into different zones, and the first zone you have to understand is A zone or A band. Now look here, I've already told you there are thin filaments, actin filaments, and there are thick myosin filaments. Now the portion inside the sarcomere that contains all the thick filament, the complete length of the thick filament, and the actin filament overlapping the myosin filament. In the diagram you can see we have these red thick filaments, and over the thick filament we have the actin filament, and the length of the actin filament that is overlapping the myosin filaments here and here. This whole region is called as A band. Now A band stands for anisotropic, and this A band, because of the overlapping, because it has the thick filament and the overlapping thin filament, it gives the dark appearance under the microscope. So that is this A band. Now moving forward, we have another band that is called as I band. Okay, now I band is only the thin filaments. You can see here is the Z line, and surrounding the Z line on both the sides we have only the actin filaments. You can see these are the actin filaments on this side, these are the actin filaments on this side. So there are no myosin filaments and there is no overlapping of the filaments. So all these thin filaments under the microscope give the lighter appearance. So we call this zone as the I band, and I stands for isotropic band. So isotropic means actually it gives the lighter appearance under the microscope. So this is called as this A band is called as the dark band as well, and this is called as the light band. Now we have another zone here that is called as H zone. Now you can see here H zone is containing the myosin filament. Now this is different from the A band. It is definitely the part of A band, but you can see here what is the difference from the A band. This contains the myosin filaments but not the overlapping actin filaments. It just contains the myosin filaments. It is the dark portion but not as dark as the A band. Okay, so it is little lighter as compared to the complete A band. So this is called as H zone, and H stands for, in German, H has been taken from the word hell. It is not that hell. This hell in German means lighter. So under the microscope, this zone was lighter, a slight lighter as compared to this whole A band. So it was given the name H band. Okay. So that actually marks the end of all these bands of the sarcomere. So let's quickly revise. We have the muscle belly. Inside the muscle belly, we have the bundles of muscle fibers. We call them as fascicles. Inside the muscle fiber, we have the myofibrils, the rod-shaped structures. Inside the myofibrils, we have the structural and the functional unit of the myofibril that is called as sarcomere, and inside the sarcomere we have the myofilaments, that is the actin and myosin. We have seen in the sarcomere has the boundaries that is called as the Z line. We have seen the A band that contains the myosin filaments with overlapping actin filaments. We have seen the I band that contains only the actin filaments. We have seen the H band that contains only the myosin filaments. And this H band is in the resting position, because if we see in the previous slide, this H zone, it is in the resting position. But once there is the contraction, all these actin filaments will be pulled. These Z lines will be pulled close to each other, and they will be pulled. That means this H zone will not appear during the contraction phase, because these actin molecules, these actin filaments, they will come close to each other. So that will decrease this H zone. This H zone is only in the relaxation phase, and in the relaxation phase, there are only the myosin here in the H zone, not the overlapping actin filaments. So that is all about the structure of skeletal muscle. I hope it made sense and you understand it. If you have any queries, or if you want any special lecture to be uploaded on YouTube, please let us know in the comment section. See you then. Thank you. Bye bye.